Shape-Specific Molecular Interaction and Binding Events, SSM-IBE. The intrinsic properties of organic molecules and their potential to fit together in a complementary fashion can be seen repeatedly throughout the biosphere. These interactions govern the inner workings of cells, the cooperation between cells during development, and the reproduction of complexity in each generation. Let's give these interactions a more descriptive name. Let's call them Shape-Specific Molecular Interaction and Binding Events, or SSM-IBE for short. When coding metaphors are replaced with SSM-IBE, it's easier to understand how complex organization can evolve. The implications of SSM-IBE for the evolution of complexity. At the instant that two or more macromolecules bind together, they form a higher-level structural unit. This is an intrinsic emergent property of molecules in general. Their ability to bind together specifically, as well as any changes that may take place as a consequence of that binding, including shape changes, as well as making or breaking strong chemical bonds, represents the structural foundation of the functional roles that they can play. In aqueous, water-based solutions, the specific shapes, binding potential, and enzymatic potential of macromolecules are set by the laws of chemistry and physics. Biochemists and molecular biologists routinely rely on these laws, in a quantitative fashion, to predict the events that may or may not occur. There is nothing subjective about any of this. The laws of nature are the same throughout the universe and are predictable by well-established principles of chemistry, physics, and biology. What this means for evolution is that SSM-IBE defines the specific organized structures and functions that can arise. Fortunately for us and other complex life forms, the potential diversity of complementary shapes and interactions among protein chains and other macromolecules is astronomical. To see this, just consider the theoretical diversity of a random sequence of 20 possible amino acids. A short random chain of just 10 amino acids has a potential diversity of 20 raised to the 10th power, or over 10 trillion possible sequences, and each has a unique shape and physical and chemical properties. These shapes become highly specific when longer protein chains fold into characteristic 3D structures, and when multiple folded structures self-assemble into more complex 3D structures. Sequences of 200 or more amino acids are quite common in protein chains, and their theoretical diversity far exceeds the number of atoms in the known universe. Metaphors implying active choices have outlived their usefulness. Human beings translate a code to something that has a clear or plain meaning by using some sort of map or table, but the biological process that we refer to as translation does not use any map or table whatsoever. Therefore, it is not actually a code at all, but rather yet another example of the emergent properties of organic molecules and their potential to interact. Is this just nitpicking? Not at all. The difference is a crucial one because it demonstrates the clear distinction between information and SSM-IBE. Information is something that intelligent beings exchange. For the purists among you, the term information also has other scientific and technical definitions related to entropy, signals, and noise that can be expressed in mathematical terms, as described by Claude Shannon and others. But those definitions of information do not provide the useful insights into the evolution of development that SSM-IBE does.